Hello everyone, today we are going to do a relatively new experiment, CUT&Tag. This method has been widely used in recent years for studying protein-DNA interactions. The entire workflow is not that complicated, so today we will follow the protocol step by step together. Today we'll follow the instructions all the way through from buffer preparation to library preparation. And finally, we can have the libraries ready for sequencing. Today we are using the Vizyme TD904 kit, which is specifically designed for the CUT&Tag experiment. It can be used to study both histone modification and transcription factors. Please know your target protein before the experiment, and the box too should be stored on ice. Besides the CUT&Tag kit, we also need to prepare other materials. We need to prepare reagents such as the primary antibody, secondary antibody, EDTA-free protease inhibitor, index primer, cleanup beads, absolute ethanol, 80% ethanol prepared later, and double distilled water. We also need to prepare some instruments and consumables, including low absorption 1.5 ml EP tubes and 8-strip PCR tubes, pipette tips, magnetic rack, ice block, vortex mixer, mini centrifuge, rotator, PCR instrument, and a high-speed centrifuge. Here is the manual, and all the information you need about this experiment can be found here. Please read it carefully before starting the experiment. In this experiment, 4 samples are used. You can adjust the number of samples as needed. The whole CUT&Tag experiment takes about 1.5 days. On day 1, we need to prepare 3 buffers: binding buffer, wash buffer, and antibody buffer. Before the buffer preparation, we need to form 10-fold binding buffer, 10-fold wash buffer, antibody buffer, and 5% dictonium at room temperature for later use, and mix well by vortexing before use. Now we are going to prepare binding buffer. The binding buffer contains divalent metal cations and is used to activate CONG-ABs Pro. Now we are going to prepare wash buffer. The wash buffer provides a stable buffered environment for cells. Please adjust the volume of protease inhibitor according to its stock concentration. Now we are going to prepare antibody buffer. The antibody buffer provides a buffered environment for primary antibody incubation, and the antibody buffer needs to be pre-cooled on ice. Now we can move on to the second step, CONG-ABs Pro preparation. For multiple samples, we can process the beads together. For today's experiment, we're going to use 4 samples. Before use, we should equilibrate CONG-ABs Pro to room temperature. Now we mix the beads well by pipetting, and please avoid rigorous pipetting or vortexing. Transfer 40 microliters of CONG-ABs Pro to a 1.5 ml EP tube. Then add 400 microliters of binding buffer and mix well by pipetting. Place the EP tube on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. To discard the supernatant completely, we can use a tip-on-tip setup. Remove the EP tube from the magnetic rack. Transfer 400 microliter of binding buffer to the tube and mix well by pipetting. Place the EP tube on the magnetic rack for about 2 minutes until the solution is clear. Transfer 40 microliter of binding buffer to the EP tube and mix well by pipetting. Alloquat 10 microliter into each well of an 8-strip tube for later use. The TD904 kit is suitable for an input range of 10 to 100,000 cells. Cell viability should be above 90%. Mix 5 microliter of cell suspension with 5 microliter of 0.4% Typen blue at a 1:1 ratio. We need to ensure the cell viability should be over 90%, so now we use a cell counter to assess cell number and viability. Transfer the required number of cells or nuclei into a 1.5 ml EP tube. Centrifuge at 600 gravity for 5 minutes at room temperature. Now we discard the supernatant. Add 500 microliter of wash buffer and pipette to resuspend the cells. Centrifuge at 600 gravity for 5 minutes at room temperature. Now we discard the supernatant. Add 400 microliter of wash buffer to resuspend the cells. CUT&Tags can bind and immobilize cells or nuclei, reduce cell loss, and enable quick separation using a magnetic rack. This is the tube containing processed CUT&Tags Pro, and now we transfer 100 microliter of cells to the tube. Mix well by inversion. Incubate at room temperature for 10 minutes. Invert 2 to 3 times during incubation. Do not pipette or vortex after CUT&Tags bind to cells. Briefly centrifuge. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Add 50 microliter of pre-cooled antibody buffer to the sample. Primary antibodies of cheap grade is recommended for this experiment. So now we add an appropriate amount of antibody to the 8-strip tube and mix well by inversion. Briefly centrifuge. Now we need to incubate the samples overnight at 4 Celsius degrees. And that is all for day 1's experiment. We can also store the remaining wash buffer here for use the next day. Hello everyone, we'll start day 2's experiment with buffer preparation. Now we are going to prepare DIG wash buffer. DIG wash buffer provides a buffered environment for cells and permeabilizes the cell membrane. Now we are going to prepare DIG 300 buffer. DIG 300 buffer contains 300 millimolar sodium chloride, which promotes chromatin condensation and inhibits non-specific cleavage by TN5 transposon. Now we are going to prepare 1-fold BNW buffer. 1-fold BNW buffer is used to activate DNA extract Bs Pro and can wash away impurities during DNA extraction. Please choose the secondary antibody based on the species of the primary antibody and make sure it has no HRP or other modifications. Transfer 200 microliter of DIG wash buffer to a 1.5 milliliter EP tube. Then add 2 microliter of secondary antibody at a 1:100 ratio. And mix well by vortexing. Briefly centrifuge and place on the magnetic rack for 2 minutes until the solution is clear. Now we discard all the supernatant. We will use the tip-on-tip thing again here for complete removal. Remove the 8-strip tube from the magnetic rack. Add 50 microliter of the prepared secondary antibody. Mix well by inversion. Incubate at room temperature with rotation for 1 hour. To save time, we can prepare the PAG-TNP Pro mix in advance during incubation and keep it on ice. Transfer 8 microliter of PAG-TNP Pro and 392 microliter of DIG 300 buffer to a 1.5 milliliter EP tube and mix well by pipetting. Place on ice for later use. Now the incubation is over, we can remove the 8-strip tube. Briefly centrifuge to collect the reaction solution. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Transfer 200 microliter of DIG wash buffer to each sample well. Mix well by inversion. Briefly centrifuge to collect the reaction solution. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Repeat the wash step 2 more times for a total of 3 washes. Now we can add the prepared PAG-TNP Pro mix and move on to the next step, PAG-TNP Pro incubation. Add 100 microliter of the prepared PAGT&P Pro. Mix well by inversion. Incubate at room temperature with rotation for 1 hour. While waiting, we can prepare TTBL buffer in advance and keep it on ice for later use. Transfer 40 microliter of 5-fold TTBL to a 1.5 ml EP2. Then add 160 microliter of DIC 300 buffer and mix well by pipetting. Place on ice for later use. Now the incubation is over, we can remove the 8-strip tube. Briefly centrifuge to collect the reaction solution. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Transfer 200 microliter of DIC 300 buffer to each sample well. Mix well by inversion. Briefly centrifuge to collect the reaction solution. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Repeat the wash step 2 more times for a total of 3 washes. Now we can add the TTBL buffer prepared earlier and move on to the next step, fragmentation. Add 50 microliter of the prepared TTBL. Mix well by inversion. Briefly centrifuge. Now we need to put the tube into the PCR instrument. Also, PAGT&P Pro is heat sensitive. Please make sure to turn off the heated lid. While waiting, we can dilute DNA spike-in according to cell inputs and target protein and keep it on ice for later use. Transfer 49 microliter of double distilled water to a 1.5 ml EP2. Transfer 1 microliter of DNA spike-in to the EP2. Mix well by vortexing. Transfer 99 microliter of double distilled water to a new tube. And then transfer 1 microliter of the solution from step 1 to the tube. Mix well by vortexing. Transfer 4 microliter of the solution from step 2 to a new EP2. Add 8 microliter of 10% SDS and mix well by pipetting. Now we remove the 8-strip tube from the PCR instrument. Transfer 3 microliter of the solution from step 3 to each sample well and mix well by inversion. Place the tube in the PCR instrument. Incubate at 55 Celsius degree for 10 minutes. Invert 2 to 3 times during incubation. Remove the tube from the PCR instrument and centrifuge briefly. Place on the magnetic rack for 2 minutes until the solution is clear. Now we transfer 50 microliter of the supernatant to a new 8-strip tube. Now we discard the beads. In this step, the storage buffer is replaced with the working buffer. We can do this during incubation to save more time. Please equilibrate the DNA extract beads Pro to room temperature before use. For multiple samples, we can process the beads together. Pipette to resuspend the beads. Transfer 100 microliter of extract beads Pro to a 1.5 ml EP2. Then add 800 microliter of 1-fold B&W buffer and mix well by pipetting. Place the EP tube on the magnetic rack. After the solution is clear, discard all the supernatant. Remove the EP tube from the magnetic rack. Add 800 microliter of 1-fold B&W buffer. Mix well by pipetting. Place the EP tube on the magnetic rack. After the solution is clear, discard all the supernatant. Transfer 200 microliter of 2-fold B&W buffer to the tube and pipette to resuspend the beads. Now we move on to step 10, DNA extraction. Transfer 50 microliter of the processed DNA extract beads Pro to the fragmentation product and mix well by pipetting. Incubate at room temperature for 20 minutes. Invert 2 to 3 times during incubation. Briefly centrifuge to collect the reaction solution. Now we discard the supernatant. Transfer 200 microliter of 1-fold B&W buffer to the H-strip tube. Incubate at room temperature for 30 seconds. Then carefully discard the supernatant. And repeat the wash step again. Air dry the beads for 2 to 5 minutes until there's no liquid residue in the tube and the bead surface becomes matte. Please note that insufficient drying or over-drying of the beads will affect library yield. Remove the H-strip tube from the magnetic rack. Add 15 microliter of double distilled water to resuspend the beads. After extraction, the DNA remains bound to the beads, so do not discard the beads. Take 25 microliter of 2-fold CAM to each well. Take 5 microliter of N5 and 4 different N7 primers to each well. Transfer 15 microliter of extraction products to each well and mix well by pipetting. Now we put the tube into the PCR instrument. Normally, we can directly proceed to purification, but if you are not sure about the cycles, you can check the concentration before purification, and you can also return the low concentration products to the PCR instrument for additional amplification. In today's experiment, we will just proceed directly to purification. Please equilibrate the DNA clean beans to room temperature before use. Mix well by vortexing. Transfer 100 microliter of the beads to the PCR product. Mix well by vortexing. Incubate at room temperature for 5 minutes. Briefly centrifuge to collect the reaction solution. Place on the magnetic rack for 2 minutes until the solution is clear. Now we discard the supernatant. Keep the H-strip tube on the magnetic rack. Add 200 microliter of freshly prepared 80% ethanol. Incubate at room temperature for 30 seconds. Discard the supernatant. Repeat this step once. Keep the H-strip tube on the magnetic rack. Air dry the beads for 3 to 5 minutes until the bead surface becomes matte. Add 22 microliter of double distilled water and mix well by pipetting. Incubate at room temperature for 5 minutes. Briefly centrifuge. Place the H-strip tube on the magnetic rack. After the solution is clear, transfer 20 microliter of the supernatant to a new EP2. Library should be stored at -30 to -15 Celsius degree. Before sequencing, we should first perform quality control, including library concentration and fragment size distribution, to ensure the libraries meet the requirements of sequencing. Please equilibrate the EcoBit reagent to room temperature before use. Add 190 microliter of working solution to S1 and S2, and 199 microliter of working solution to each sample tube. Add 10 microliter of standard 1 to S1 and 10 microliter of standard 2 to S2. Add 1 microliter of each library sample into the corresponding sample tubes. Mix well by vortexing. Place S1 into the instrument and read standard 1. Then remove S1, insert S2, and read standard 2 to generate the standard curve. Then we can put the samples into the instrument. Equilibrate the adjuvant reagents to room temperature before use. Add 9 microliter of gel into the G well. Add 9 microliter of gel into the G well. Add 5 microliter of marker and 1 microliter of ladder into the ladder well. The total volume in each sample well is 6 microliter. The exact volumes can be adjusted based on sample concentration. In this experiment, add 4 microliter of marker and 2 microliter of library to each sample well. Centrifuge for 1 minute. Select the DNA 1000 assay program and click Start. So the whole experiment is now over. Thank you for watching this video. We really hope it is helpful to your own experiment.